Several factors can affect the progression of Huntington's disease. This video will explain recent research into a DNA repair protein called FAN1 and how a small change in this protein can alter HD progression. HD is caused by a mutation in the Huntington gene. Everyone has a repetitive sequence of CAG DNA letters in their HTT gene. But people who develop HD have over 40 CAG repeats. Longer CAG repeats are associated with an earlier onset of HD symptoms and faster disease progression. However, other factors known as genetic modifiers can also affect when HD symptoms start. Genetic modifiers are proteins that have changes to their code, which makes them more or less likely to speed up or slow down disease progression. Proteins are made up from building blocks known as amino acids, and genetic mutations can cause changes to the sequence of protein blocks. Everyone has small genetic differences in the code making up the proteins in their body, which normally do not affect a protein's function. However, in some cases, a mutation may alter the function of a protein, which could lead to earlier or later onset of a disease. These proteins are called genetic modifiers. Several DNA repair proteins were discovered to be genetic modifiers of HD. DNA repair proteins constantly keep our genetic material in check and correct harmful errors. This is extremely important, as our DNA is under constant stress and gets damaged under several circumstances, including exposure to UV light from the sun. Molecular processes in your cells can be prone to errors, and DNA repair ensures no harmful mutations occur. DNA repair has been found to be particularly important in HD. This is because researchers have shown that in HD, the repeating CAG DNA region of the HTT gene increases in length over time in a process called somatic expansion. Somatic expansion is thought to heavily influence HD disease progression and severity. Some DNA repair proteins have been shown to play a big role in managing these repetitive DNA regions and affect somatic expansion of the CAG repeat. Specific genetic mutations in the code of these DNA repair proteins have been associated with accelerating or delaying the age at which HD symptoms start. In this video, we will focus on one key DNA repair player called FAN1. Some people with HD carry a specific change in FAN1 which either speeds up or slows down the progression of HD. One specific mutation in FAN1 is called R507H. The R in this R507H represents a protein building block at the 507th position in the protein sequence. The genetic mutation causes the R building block to be swapped with an H, which is why there is a H at the end of the name. Although this is a small change, people with this FAN1 variant tend to develop symptoms much earlier than expected. Researchers have studied how the R507H mutation in FAN1 affects its function. Using high-powered microscopes, researchers were able to study how different forms of FAN1, mutant and non-mutant, affected its ability to bind to DNA and carry out its DNA repair activity. The researchers found that FAN1 with the R507H mutation bound less tightly to other DNA repair proteins, such as pCNA, suggesting DNA repair was not being performed as effectively. Another study investigated the effect of the R507H mutation on FAN1's ability to cut loops of DNA that form at repetitive DNA regions like long CAG repeats in the HTT gene. These DNA loops can lead to further CAG expansions, worsening HD symptoms over time, and so it is important that they are repaired correctly. Importantly, the R507H FAN1 was worse at cutting the loops in comparison to the non-mutated FAN1. These findings suggest why people with HD who have the R507H mutation might develop symptoms earlier, as this mutation deteriorates FAN1's DNA repair activity by both reducing its interaction with pCNA and worsening FAN1's ability to process loops of DNA. This may lead to a buildup of harmful changes to the HTT DNA sequence, increasing the risk of somatic expansion. This research has increased our understanding of HD and has also highlighted the possibility of using FAN1 as a therapeutic target for HD. If we could enhance the repair function of FAN1 or stabilize the FAN1/PCNA interaction, this may slow somatic expansion of the CAG repeat, which could slow disease progression. One thing to note is that these experiments were not performed in living systems, so further experiments will need to be carried out to confirm that enhancing FAN1 function would be healthy for HD patients. Most of the current clinical trials are attempting to reduce Huntington protein levels in patients using a whole host of therapeutic techniques. This research has been very exciting for the field. However, it is great that we have alternative therapeutic avenues to explore as a future therapy for HD.